Can someone kindly unmute and tell me if I'm audible and if the screen is visible?
Yes, ma'am.
Okay, okay, thank you so much. Okay, so we'll start today's session. Good evening, everyone. My name is Aishi and I am a PMRF research scholar from the Department of Chemistry, IIT Bombay. And I'm your PMRF TA for this course, Applied Environmental Microbiology. Today, we'll have our third live session for this course, and we'll also discuss some questions, some relevant questions. Hopefully, this will help you to solve this year's current assignments as well. So, we'll move to the first question. Anaerobic utilization of glucose happens in fermentation. So all these questions are based on the lecture slides from the NPTEL course. So anybody would like to give this a try, you can unmute and let me know.
True.
Okay. Anybody else has any kind of conflicting opinion, or do you all agree? So yes, the answer is true. So we'll discuss a little bit about what happens during fermentation. So, but before that, we need to know what anaerobic utilization of glucose means. So, the term anaerobic basically stands for absence of air or free oxygen. And accordingly, anaerobic respiration is a type of cellular respiration in which the stored energy of glucose gets converted into biochemical energy coins, ATP, without any free oxygen. And many microorganisms rely on this process to produce energy when there is a lack of oxygen. So these microorganisms can include prokaryotes like bacteria, archaea, and can even include eukaryotes. So many microorganisms then use nitrate and sulfate as electron acceptors, and then they reduce it to nitrite or hydrogen sulfide respectively. And anaerobic respiration exclusively occurs in the cell cytoplasm. So similar to aerobic respiration, anaerobic respiration also starts with glycolysis, which is the first step, which means in this step you convert glucose into pyruvate. So if you can see the screen over here, the first step is glycolysis, and you convert glucose into pyruvate, and where ATP is also produced. And but the pyruvate does not further enter the mitochondria to complete the aerobic respiration due to the absence of oxygen. So in aerobic respiration, this pyruvate will further enter the mitochondria to carry forward with Krebs cycle and then oxidative phosphorylation. But here in this case, the pyruvate does not enter the mitochondria to complete the aerobic respiration because obviously there is no oxygen around. So instead, what it does, it follows a different pathway altogether and gets converted into a different byproduct in the cytosol only. So this is a rough sketch of the mechanism. So what are the steps of the pathway? As I said, it occurs through glycolysis and then fermentation. And glycolysis is the first and common step of both the types of respiration. And one glucose molecule splits into two pyruvate molecules, releasing electrons in the process, and also it generates ATP. And in this, it's a huge 10-step biochemical pathway. There's a lot of enzymes involved that has not been shown in this sketch. I think I had showed in one of my previous slides. So in this 10-step biochemical pathway, 4 ATP molecules are formed by substrate level phosphorylation. Among them, 2 ATP molecules are then consumed during the initial steps. So hence the net yield of ATP from glycolysis is 2 ATP per glucose molecule. And additionally, 4 electrons are released during the formation of NADPH, which can then be used to produce ATP by aerobic respiration. But when it moves when anaerobic respiration is happening, it will move into fermentation. So what happens, due to the absence of oxygen, the electron transport chain, or ETC, it fails to function as O2 is not present and as the final electron acceptor. If you remember, in electron transport chain, the oxygen was the final electron receptor, but in this case that is not possible. So the pyruvate that is produced in the glycolysis, that is in the previous step, it cannot enter the mitochondria for aerobic respiration. As a result, there is an excess of NADH and pyruvate in the cytosol. As glycolysis is the only energy yielding phase of anaerobic respiration, it needs to be repeated frequently to produce ATPs within a short period. So what was happening in aerobic respiration? There was Krebs cycle and ETC, electron transport chain, and total there was 38 ATP molecules produced. But here in this case, since glycolysis is the only mechanism which gives ATP, which gives energy, so this process needs to happen quite frequently to produce ATPs within a short period, and thus glycolysis runs ceaselessly if a continuous supply of NAD plus is available. And to fulfill that demand, the NADH that is produced in the previous step donates its electrons to pyruvate and converts into NAD plus, producing organic acid or alcohol as a byproduct. And this phenomenon of producing acid or alcohol from pyruvate by the action of NADH is referred to as fermentation. So we can have there's a common question: how many ATPs are produced during anaerobic respiration? So in anaerobic respiration, 2 molecules of ATP are produced from 1 molecule of glucose. So there are two types of fermentation. One can be alcoholic fermentation, and the other can be lactic acid fermentation. In living organisms, these are the two types of anaerobic respiration that can happen. So in the lactic acid fermentation, what happens? The NADH transfers its electrons directly to the pyruvate, and it generates lactate as a byproduct. And lactate is just the deprotonated form of lactic acid, which gives the process its name. That's why it's called lactic acid fermentation. And the lactate that is produced is cycled out of the cell and then transported into the blood, and a part of it goes to the liver and converts back to glucose during the Cori-Cori cycle. So glucose is because glucose is essential for the cells. And this type of fermentation is generally used in mammalian, like happens in mammalian red blood cells and skeletal muscle with insufficient oxygen supply. So muscle cells at that time use an enzyme called the lactate dehydrogenase for this process. The other process is the alcoholic fermentation. In this process, what happens? The NADH donates its electrons to a pyruvate derivative. In lactic acid, it was donating directly to pyruvate, but in alcoholic fermentation, it basically donates the electrons to a pyruvate derivative, producing ethanol in 2 steps. And in the initial step, a carboxyl group is removed from pyruvate and released as carbon dioxide, producing a 2-carbon molecule called acetaldehyde. And in the second step, what NADH does is that it passes its electrons to acetaldehyde, regenerating NAD plus and forming ethanol or ethyl alcohol. And this process is called alcoholic fermentation or ethanol fermentation, as the final product of this reaction is ethanol. So this process is conducted by microscopic organisms such as bacteria and some fungi such as yeast. So these are the two processes. This schematic shows the two processes in very much detail. As you can see, in lactic acid fermentation, what's happening? And lactic acid fermentation can happen in muscle cells when we are running, and after some point of time our muscle cells start aching. That's because there's an accumulation of lactate that is created during this part of anaerobic respiration. So this is if the aerobic, so the red part is when aerobic respiration is happening, O2 is present, and you can see glycolysis happens, pyruvate forms, it goes into mitochondria, then moves ahead to Krebs cycle. But when O2 is not there, the lactic acid fermentation pathway happens. In case of ethanol fermentation, you can see aerobic respiration, if not there, then acetaldehyde is formed. So alcoholic fermentation occurs in 2 steps. Acetaldehyde is formed and then ethyl alcohol or ethanol is formed. So you can see this is a very zoomed in image of the lactic acid fermentation, and this is alcoholic fermentation when O2 is absent. So are there any questions regarding this? Because the process is kind of similar, both lactic acid and alcoholic fermentation. It's just that in lactic acid fermentation, NADH directly transfers electrons to pyruvate, but in alcoholic fermentation, there is a pyruvate derivative that comes in between. So these are the two processes of fermentation. So the question was, anaerobic utilization of glucose happens in fermentation, which is true. Anaerobic respiration does happen, and the process is called fermentation. So the next question is, SO4 minus can be used as an electron acceptor in anaerobic respiration by some facultative bacteria. Anyone? Oh, one more thing I forgot to mention in the last question. If you're talking about the advantages of anaerobic respiration, like obviously there are a lot of advantages of aerobic respiration, but if you're talking about anaerobic respiration and some advantages, I'm still in the meeting, right? Yeah. Advantages of anaerobic respiration: it is a relatively rapid process because ATP is produced very quickly. For instance, it allows muscles to get the energy they need for short bursts of intense activity. So it is a quick process. Also, it allows organisms to live in places with little or no oxygen, such as deep water and the digestive tracts of animals like humans. We have a very diverse microbiota, gut microbiota, if you're aware. And it can also act as a backup when cellular oxygen is depleted in multicellular organisms that require oxygen, such as human beings. And it can also assist aerobic respiration by metabolizing pyruvic acid, the end product of glycolysis, and in this process regenerating enzymes necessary for glycolysis, and this facilitates further aerobic respiration. So these are some of the advantages of anaerobic respiration. And if you're talking about which organism is most likely to go through this process, I think yeast is one of the organisms that goes through this process. It's very likely to use anaerobic respiration. and the rate of anaerobic respiration generally increases with increasing temperature. And so fermentation is the process, and the difference in anaerobic respiration in plants and animals is that in plants the end products are ethanol and CO2. In animals, the end product is lactic acid. As you can must have felt when you're running for a long period of time, your muscles start aching. That's because of this lactic acid fermentation, formation, accumulation in the muscles. 1 second, I'll accept. So that is one of the differences between anaerobic respiration in plants and animals. Also, in plants, the accumulation of ethanol may kill the plant. In animals, the lactic acid does not cause death, but may lead to minor muscle cramps that can be cured with hot water bath or a massage. And in plants, CO2 gets released, but in animals, CO2 is not released. And like, when do humans respire anaerobically? If someone asks, in humans, anaerobic respiration mostly occurs in muscles during high intensity exercise when there is less oxygen supply. And obviously, anaerobic respiration is much less efficient than aerobic respiration, because we know in aerobic respiration, the entire process, glycolysis, Krebs cycle, etc., or the electron transport chain, all in all, they generate 38 ATPs. But here it's only 2 ATPs. So you can figure out the difference. That's why aerobic respiration is much more efficient and energy yielding. So that's it. So next question was SO4 2 minus can be used as an electron acceptor in anaerobic respiration by some facultative bacteria. Anyone?
True.
Okay. So yes, that is true, because in case of anaerobic respiration, basically the process is formation of ATP without oxygen, as I discussed right now. So this method but still incorporates the respiratory electron transport chain, but it obviously doesn't have oxygen as the terminal electron receptor, as we see in ETC, or the electron transport chain during aerobic respiration. But instead, molecules such as sulfate, nitrate, or sulfur are used as electron acceptors, and these molecules have a lower reduction potential than oxygen. Thus, less energy is performed per molecule of glucose in anaerobic versus aerobic respiration. So that's pretty much what I explained in the last slide only. That they can use molecules such as sulfate, nitrate, and sulfur. Someone was okay. Sulfur and 1 second. Sorry guys for the interruption. There's this. Yes. So this sulfate reduction basically happens when sulfate is used as the electron acceptor, producing hydrogen sulfide as a metabolic end product, and is relatively an energetically poor process, as I already explained why, and is used by many gram-negative bacteria found within the gamma proteobacteria, and it is also used in gram-positive organisms also, related to this desulfotomaculum or the archeon archeoglobulus. These are very factual questions, so if you want to learn more about it, maybe you can search which organisms are more likely to use sulfate as the electron acceptor. And this sulfate reduction uses electron donors such as the carbon compounds lactate and pyruvate, or hydrogen gas in case of lithotropic reducers, and some unusual autotrophic sulfate reducing bacteria also there. These are some of the scientific names. And they can also use phosphite as an electron donor. Others, such as certain disulfovibrio species, these are capable of sulfur disproportionation, that is splitting one compound into an electron donor and an electron acceptor, and using elemental sulfur, sulfite, and thiosulfate to produce both hydrogen sulfide and sulfate. So yes, so as we know, many different types of electron acceptors may be used for anaerobic respiration, and such as denitrification is the technique where nitrate is used as a terminal electron acceptor, and nitrate, like oxygen, has a high reduction potential, and this process is really widespread and it's used by many members of proteobacteria. And many denitrifying bacteria can also use ferric iron, that is Fe3 plus, and different organic electron acceptors. So sulfate reduction is also similar to that. And then acidogenesis is a type of microbial metabolism that uses hydrogen H2 as an electron donor and carbon dioxide as an electron acceptor to produce acetate. The same electron donors and acceptors are used in methanogenesis also. Then there is use of ferric iron, or Fe3 plus. It's also a very widespread anaerobic terminal electron acceptor, and it's used by both autotrophic and heterotrophic organisms. And electron flow in these organisms is quite similar to those in electron transport, ending in oxygen or nitrate, except that in ferric iron reducing organisms, the final enzyme would be a ferric iron reductase. And since some ferric iron reducing bacteria can use toxic hydrocarbons such as toluene, as a carbon source. There's a significant interest, actually, basically it's fascinating because people are interested in these organisms because as bioremediation agents in ferric iron contaminated aquifers, they can actually use such bacteria which can use toxic hydrocarbons like toluene. And there are some other electron acceptors also in organics, such as I think manganic ion, then manganese Mn2 plus, manganic is Mn4 plus, manganese Mn2 plus, selenate, selenite which converts to selenite, then selenium, then arsenate to arsenite, and there's so many. And organic compounds are also sometimes can also be used as electron acceptors in anaerobic respiration, and these include the reduction of fumarate to succinate, then trimethylamine N oxide to trimethylamine, and dimethyl sulfoxide, which is DMSO, to dimethyl sulfide. So these are some of the common examples of organic compounds also which can also be used. And sulfate reduction, it's a major anion in seawater, and this is one of the sulfate reducing bacteria, and all sulfate reducers are bacteria only. And Archiglobus is the only archaea, like from the prokaryote domain, there's bacteria, most sulfate reducers are bacteria, and amongst the Archiglobus is the only archaea that can do sulfate reduction. And this shows the biochemistry of sulfate reduction. What happens, there are two types, dissimilative sulfate reduction and assimilative sulfate reduction. When sulfate is basically used for some kind of purpose, that's called assimilative. And there are organic sulfur compounds such as cysteine, methionine, these are all amino acids that can be used, and this is another dissimilative sulfate reduction. And in sulfate reducing bacteria, this is the electron transport and energy conservation that is happening. And in addition to the external H2 is also originating from the catabolism of organic compounds. So you can see how the entire process is going on. So the question was, sulfate can be used in electron acceptor in anaerobic respiration, and the answer is true. Yeah, she was correct. Move on to the next question. Oxygenic photosynthesis requires carbon dioxide. Anyone? Anyone would like to try?
True.
True. Yes. So as we all know, photosynthesis is basically the process that is used by plants, algae and some bacteria to turn sunlight into energy. And this process chemically converts carbon dioxide from the environment and water into food, sugars and oxygen. And this chemical reaction often relies on a pigment called chlorophyll, which gives plants their green colors basically. And photosynthesis is also the reason of that our planet is blanketed in oxygen-rich atmosphere, because obviously plants release oxygen, and that's what we are breathing in every second. So there are two types of photosynthesis, oxygenic and anoxygenic, and the names are pretty much self-explanatory. So they both follow very similar principles, but the former is the most common and seen in plants, algae and cyanobacteria. So these are the equations for oxygenic and anoxygenic photosynthesis, as you can see from the slide. So during oxygenic photosynthesis, what happens is that light energy comes and it is transferred. Basically, the light energy is transferred. This transfers electrons from water, and that is taken up by the plant roots from the soil to CO2, and this produces carbohydrates. And in this transfer, the CO2 gets reduced. The CO2 that was the starting material gets reduced or receives electrons, and the water is oxidized. The water gets oxidized or loses electrons, and oxygen is produced along with carbohydrates. And this process basically creates a balance on Earth, in which the carbon dioxide produced by breathing organisms as they consume oxygen in respiration is converted back into oxygen by plants, algae and bacteria. So we breathe in oxygen, we give out carbon dioxide. So at this level, there should be no oxygen present in the atmosphere, but it's only because of the trees who give out oxygen in return, and they consume CO2, and that's why we have oxygen from the air that we breathe in. Apart from oxygenic photosynthesis, there is anoxygenic photosynthesis, which uses electron donors that are not water, and the process does not generate oxygen. This process typically occurs in bacteria such as green sulfur bacteria and phototropic purple bacteria. So you can see here, the water is not the electron donor, it's H2A. H2A stands for any electron donor. It can be hydrogen sulfide H2, but basically water is, it's not H2. Water is the electron donor is not the water basically. So these are some of the differences between oxygenic and anoxygenic photosynthesis. So oxygenic photosynthesis is the photosynthesis that occurs in plants. It's the most common ones that we study in school. It's in the final electron accepted is water. There is oxygen produced, there is carbohydrates produced, and CO2 is taken in, it's consumed. In anoxygenic photosynthesis, it's used by certain bacteria obviously, and oxygen is not produced. That's quite self-explanatory from the name. Where does it occur? The oxygenic one occurs in plants, algae, and cyanobacteria. For the anoxygenic one, it occurs in green sulfur and non-sulfur bacteria, purple bacteria, helicobacteria, acidobacteria, all these. Then both photosystem 1 and 2 are used in case of oxygenic photosynthesis, but in case of anoxygenic, only photosystem 1 is used. And in case of oxygenic, water is the electron source, as we saw in the last slide. But in case of an anoxygenic, it will be H2 or H2S, or sometimes even ferrous ions can serve as the electron donor. In oxygenic, oxygen is produced during the light reaction, but in anoxygenic, that is not the case. Oxygenic chlorophylls are used. In anoxygenic, bacteriochlorophylls or chlorophylls are used. And in oxygenic, ADP or adenosine diphosphate serves as the terminal electron acceptor that produces NADPH. But in anoxygenic one, NADPH is not produced as the electrons are cycled back to the system. And in case of oxygenic, obviously ATP is produced by non-cyclic photophosphorylation. But in case of anoxygenic, it is produced by cyclic photophosphorylation. So though both types of these photosynthesis are complex, multi-step affairs, the overall process can be summarized as this chemical reaction, as I showed in the previous slide only. Here in this case, there is water as the starting material. In the anoxygenic case, water is not a starting material. It's H2. It represents the potential electron donor, can be H2S, hydrogen sulfide, or H2 as well. And we know that plants absorb CO2 from the surrounding air and release water and oxygen via these microscopic pores on their leaves called stomata. And when stomata open, they let in, they basically consume CO2. However, while open, the stomata also releases oxygen and let water vapor escape. And the stomata then close to prevent water loss, but that means the plant can no longer gain CO2 for photosynthesis. And this trade-off between CO2 gain and water loss is a particular problem for plants growing in very hot and dry environments because their water is very limited. So we'll move on to the next question. Energy tapped during energy tapped during chemoheterotrophy from inorganic molecules is more than that of glucose. More than that from glucose. Anybody?
False.
False. Correct. Obviously, no matter what the organic molecules, the energy cannot be more than that of glucose. When glucose is used, we all know what the process are. Glycolysis, where simple glucose is broken down to pyruvate. Then pyruvate is transformed to acetyl CoA in the mitochondria for the next step. Then in the mitochondria, the next step happens, which is the citric acid cycle, the TCA cycle, or the Krebs cycle, all the same. And in this case, the acetyl CoA is modified in the mitochondria to produce energy precursors in the preparation of the next step, which is oxidative phosphorylation, where the electron transport chain is happening, and electron transport happens from the energy precursors from the citric acid cycle, whatever we got from the citric acid cycle, and it leads to the phosphorylation of ADP, producing ATP, and this occurs in the mitochondria. So the entire process gives out 38 ATP molecules, which is a huge deal. So obviously, whatever inorganic molecules are broken down, energy cannot exceed that from glucose. Next question is metabolic similarity frequently maps well with functional similarity in microbes. Anybody would like to try? What do you think? Any guess is fine. Still in the meeting, right? Yeah. Anybody?
True.
Okay. Why do you think so? Is there any particular reason? Okay, so I'll explain. So this is taken from the slide of the course only. So what is functional diversity is a concept. So it only says in the slide that metabolic similarity often maps well to the functional similarity and vice versa, which is, if you think about it in a very it's common sense, like when functional similarity is happening, that means there are some metabolic processes that are tend to become similar. That is functional whatever is happening in as a function attribute, it's very similar to whatever the metabolic processes are happening inside. That's why metabolic similarity often maps really well with the functional similarity, as the functional profile is directly related to the environmental factors in the niche, thus is representative of its environment. So obviously whatever functions you're doing, whatever function any organism is doing, that is very relevant to the environmental factors, the environment that that organism is surviving in. And this is not necessarily true for all phylogenetic associations, namely phylogenetically similar microbes may be functionally dissimilar. There was this one question in one of the previous sessions that we discussed about microbes having same color being same similar kind of that was the kind of question. So even phylogenetically some similar microbes that have been phylogenetically put in a very close manner, very close, they're very close on the branch or clade in the phylogenetic tree, they may be functionally very different, they're very dissimilar, and vice versa. Due to gene loss, there can be convergent evolution, there can be horizontal gene transfer such as transformation, transduction, conjugation. So these are the three processes of horizontal gene transfer. We discussed this in the last session. So these reasons can be can be held responsible for similar phylogenetically similar microbes still showing these similar functions, but metabolic similarity generally maps very close to very well with functional similarity. And what is functional diversity then? So there can be three types. There can be physiological diversity, where functions and activities of microorganisms, microbial metabolism, and cellular biochemistry, all of these will be taken into account. There can be ecological diversity, where the relationship between the organism and its environment only is taken into account. And there can be morphological diversity, obviously the morphology of the system, what is the outward appearance, what is the countenance of the system. So these are different types of functional diversity if you're trying to relate. So the next question is, essential element for the growth of hydrogen metabolizing bacteria in culture is. This also we discussed, a similar question we discussed in the last session. Anybody? You can think about it and answer, there's no hurry. Okay, I'll answer. So it's nickel. We discussed a question like this in the last session also. So hydrogenases are basically nothing but gas processing redox enzymes, very central to the hydrogen metabolism. And nickel also participates in various metabolic processes such as acidogenesis, hydrogen metabolism, and biogenesis of methane in bacteria. So the reference of the paper is also given where these information have been discovered. The enzymes such as acetyl coenzyme A synthase, carbon monoxide dehydrogenase, hydrogenases, methyl coenzyme M reductase, then NIF hydrogenases, RNase A, urease, all of these enzymes require NIF as a catalytic partner for their catalytic functioning. And this long-term and excessive use of fertilizers containing zinc, magnesium, copper can effectively induce nickel deficiency, as these metals sometimes inhibit the uptake of nickel very competitively. That is why nickel is considered as a very essential element for a variety of microbial cellular process. So you can go through this paper by Mulrooney and Hosinger that was published in 2003. They have talked in detail regarding this process. Also, transportation of nickel into bacterial cells. One second. Transportation of nickel into bacterial cells involves ATP binding cascade. Also, we discussed about the ATP binding cascade also on the first day. ABC transport protein basically. So these transportation of nickel into these bacterial cells, this involves the ABC cascade, ATP binding cascade or ABC transporter, while nickel enters into archaea through nickel cobalt transporters. So in bacterial cells, they basically mainly use ABC transporters, but in archaea they have these transporters for their specific transporters. So this information is taken from this paper, Ittinger and Matrand Berthelot, and these two papers. So if you're interested, you can also go through these papers and read more about the importance of nickel in the growth of basically hydrogen metabolizing bacteria in culture. Which of the following is most likely not to be found in a hot spring? Anybody would like to answer? This is a very confusing question. If anybody wants to answer, they can. Anybody?
Green non-sulfur bacteria.
Why do even I thought that before at first, but this question is very confusing. Honestly, I am yet to learn more about this. I actually went on Google and searched, but there are some green non-sulfur bacteria that are found in some New Zealand hot springs or somewhere. So we need to actually research more about this on Google. So even if this question is a little outdated, there are some papers that talk about some new green non-sulfur bacteria that have been discovered from hot springs in New Zealand. So that is but why do you think, why does green non-sulfur bacteria like, why is it most likely that it will not be found in a hot spring? Do you have any reason for that? Did you think about it when you said, like, do you have any reason behind your answer? Because generally I wanted to know if there was someone having any reasoning, because this
Because it is
hmm.
it is found in on not found in hot springs mostly.
Why do you think green non-sulfur bacteria will not be found in hot springs? Is there any reason based on any kind of mechanism or anything that you thought of?
No, sorry.
Okay, okay. So yeah, so I think I read about some green non recent paper or something. I'll look into more about it, and we can discuss this question in the next session also. I think I thought a green non-sulfur bacteria, but then I think I read somewhere that green non-sulfur bacteria have also been discovered from New Zealand hot springs. And even if there are some exceptions happening, why green non-sulfur bacteria will not be present in a hot spring? Hot spring is a very thermophile, like very extreme environment. So we can discuss this question next day. So if you're in the meeting, we can actually go back and read more about it, and we can discuss, come back to this question in the next meeting. So carotenoid gives color to the bacterium. Is it true or false? It's also a very basic question. Anybody?
Yes, it is true.
Yes, correct. So carotenoids are basically tetraterpene pigments, and they exhibit different colors like yellow, orange, red, purple, and they are very widely distributed pigments in nature, and they can be found in photosynthetic bacteria also. As you can see, some of the images I've taken from a paper, and this can also be seen in some species of archaea or fungi, algae, plants, and animals. And most carotenoids exist in 8 isoprene units with a 40 carbon skeleton. So that is most about the chemistry. So this I took from one of the papers, which shows light dependent carotenoid like pigment for production of isolates and genome sequenced bacteria, and light dependent phenotype of strains grown at 28 degrees Celsius under dark and blue light conditions. Depending on strains, depending on temperature, depending on the kind of light you're exposing it to, all of this matters. You can see the difference in color as well. There's basically a gradient there, even though the colors are, the colorful ones are mostly yellow, but still you can see that there's a difference in the shade intensity, difference in gradient basically formation. So that's how. So yes, the question was, it gives color to the bacterium. Definitely it gives color to the bacterium. It's a very true statement. It's a popular pigment. Next question is that a genetic marker used for building a phylogenetic tree should be universally present in all bacteria. Anybody who would like to try?
It's false. Since
Acha, why do you think it's false?
is some bacteria genetic material is not react to any dyes.
Okay, but like, if we are building a phylogenetic tree, we should have something universally present in that. That's how we'll be able to, basically relate those. Like, if we have talked about 16S rRNA, which is very conserved, which is a very conserved element, and that is mostly used for 16S rRNA sequencing, and it is also a very popular element that is used for phylogenetic tree analysis. So in that case, if we are preparing a phylogenetic tree, no matter what organism, if we are preparing, there's something that we need to keep in mind, something universal that should be able to relate the organisms to each other. So in that case, if you think like that, the genetic marker, the this one would be true, because yes. So several characteristics of the 16S rRNA, that I'm choosing this because we talked about 16S rRNA in one of the sessions, and also it's very universally, is mostly conserved across various species. So that's why several characteristics of the 16S rRNA gene, such as essential function, ubiquity, and evolutionary properties, all of these are very relevant, and all of these have allowed it to become the most commonly used molecular marker in microbial ecology. And there are mostly the phylogenetic trees are relevant and dependent on the 16S, this universal ubiquity. So yes, if you're trying to relate, if you're trying to compare, or if you're trying to at least show the relationship between various organisms, I think one essential element is very important, that will be universally present, that will be universally expressed. So moving on to the next question, oxidative phosphorylation happens in cellular respiration. Anybody? This also we discussed at length in the previous class.
True.
True. Correct. So we know oxidative phosphorylation happens, it's basically the major energy provider of the cell. And as we know that there are a lot of different ways the organisms acquire food. Like almost all aerobic organisms that require oxygen to live, like us, use oxidative phosphorylation in one way or another to produce the basic energy currency of the cell, that is ATP, or adenosine triphosphide. And it is it is a step that produces the most of the energy in cellular respiration. So where does oxidative phosphorylation basically fit into the cellular respiration? Glycolysis I talked about, where the simple sugar is broken down to pyruvate in the cytosol. Then pyruvate, the product from glycolysis, is transformed into acetyl CoA and in the mitochondria for the next step. And then happens the citric acid cycle, or the Krebs cycle, where the acetyl CoA is further modified into in the mitochondria for to produce energy precursors in propagation for the next step. And then comes oxidative phosphorylation, which is the process where electron transfer from the energy precursors from the citric acid cycle, or the TCA cycle, leads to the phosphorylation of ADP, producing ATP, which is adenosine triphosphate. And this also occurs in the mitochondria. So as you can see from the sorry, is there anyone? Is there anyone? Okay, I'm still in the meeting. Sorry. So as you can see, oxidative phosphorylation is the process of ATP formation. And you can see the basic rough sketch over here. It's the final step. It occurs in the mitochondria. It is linked to this process called the electron transport chain. And it happens in the inner mitochondrial membrane. And the electrons are transferred from one member of the transport chain to the another through a series of redox reactions. So this is a very rough sketch of the oxidative phosphorylation. And then the electron transport chain happens. So these are some of the steps of the electron transport chain. It has 4 complexes. So the oxidative phosphorylation is the process where energy is harnessed through the series of protein complexes, basically embedded in the inner membrane of the mitochondria. And this is what leads to the ETC, the electron transport chain, and this leads to the creation of ATP. So it can be broken into 2 parts: oxidation of NADH and FADH2, and phosphorylation. So what happens in step 1? Oxidation of NADH and FADH2 occurs. So these two substances, they basically lose electrons via high energy molecule. Sorry, one second, I need to. So what happens? These two substances, they lose electrons via high energy molecules, and this oxidative phosphorylation like it commences with the arrival of 3 NADH and 1 FADH2 from the Krebs cycle, which shuttle high energy molecules to the electron transport chain, basically. And this substance is then transferred its high energy molecules to protein complex 1, while FADH2 transfers its high energy molecules to protein complex 2, as you can see over here. One second. As you can see over here. And then shuttling high energy molecules causes the loss of electrons from NADH and FADH2, leading to the formation of NAD plus and FAD. And this process is called oxidation. And the opposite of oxidation is reduction, basically, where a molecule gains electrons, and that happens in the citric acid cycle. So you can see FADH interacts with the complex 2, NADH transfers electron high energy molecules to the complex 1 in the first step. In the next step, this oxidation process leads to the pumping of protons. Protons are nothing but single positively charged hydrogen atoms, and they are denoted as H plus. So this NADH oxidation leads to the pumping of protons through this complex 1 from the matrix to the intermembrane space. So this side is the matrix. The entire electron transport chain is happening in the inner mitochondrial membrane. So this side is the matrix, the outer side is the intermembrane space, the space between the outer and the inner membrane. So the protons are pumped through this protein complex 1 from this matrix to the intermembrane space. And the electrons that were received by protein complex 1 from NADH are then given to another, relayed. Basically, these electrons are relayed to another membrane bound electron carrier called ubiquinone, or Q, denoted as Q. And this process of transferring electrons drives the pumping of protons, which is seen as unfavorable. And electron transfer driving proton pumping is repeated in complexes 3 and 4 as well, which we'll discuss in the further steps. And as this action is repeated, protons will accumulate in the intermembrane space. Obviously, protons will start accumulating in the intermembrane space. And this accumulation of protons is how the cell temporarily stores transformed energy. So this is their technique of storing the transformed energy that is getting there. And this FeDH2 has a slightly different route than NADH. After its arrival at the protein complex 2, its high energy electrons are directly transferred to Q to form reduced Q or QH2. So there is no hydrogen pumping for the exchange of FeDH2 electrons here, but there was hydrogen pumping happening for NADH. So these two go through a slightly different process. So this is what happens in step 2. In step 3, the rest of the steps are now same for both the molecules as in earlier steps. So inside the non-polar region of the phospholipid bilayer, so inside is the non-polar region, the hydrophobic inside of the phospholipid bilayer. So what happens? The reduced ubiquinone, which is also a very non-polar compound, it transports the electrons to protein complex 3, as you can see in the sketch over here. And this reduced ubiquinone also carries protons. So when this UQH2, or the reduced ubiquinone complex, delivers electrons to this protein complex 3, it also donates its protons to be pumped. As you can see from the sketch over here, protons are given out to be pumped. And then in the step 4, the electrons that arrive at the protein complex 3 are picked up by cytochrome C. And this is the last electron carrier over here. And this action also causes protons to be pumped into the intermembrane space. As you can see, this process is happening. It's a means of storing the transformed energy, as we talked about in the previous slide. So this action also causes protons to be pumped into the intermembrane space. And then comes the next step. In this case, as we have seen that cytochrome C has already received the electrons, so it will carry the electrons to the final protein complex, or protein complex 4. And once again the energy released via the electron shuttling allows for another proton to be pumped into the intermembrane space, as you can see from here. And these electrons are then drawn to, proton is pumped out, these electrons are then drawn to this oxygen, which is the terminal final electron receptor, as we talked about previously also. So oxygen is the final electron receptor. And it's very fascinating that oxygen must be present for the oxidative phosphorylation to occur. So, and as a result of which water is formed, as oxygen receives the electrons from protein complex 4 and combines with protons on the inside of the cell. So there are protons, there's a proton reservoir in the inside of the cell also. So the oxygen receives the electrons, interacts with proton, and it forms H2O. So this process can only happen in the presence of oxygen, that is in aerobic respiration. In anaerobic respiration, where oxygen is not present, this process is not possible, and that's when the cells move into fermentation. So in summary, what happens? There are 3 NADH. There's one FADH2. There are 3 hydrogen protons pumped out and pumped. There are the 2 minus 2 hydrogen protons, and then we have O2, which receives the electrons, and one H2 is formed. This is the previous steps. As we had in the first slide, we had said that we can actually divide this entire process into 2 steps: oxidation of NADH and FADH2, and the second step is phosphorylation. So the first step is done. Now the second step will start, which is phosphorylation, or the production of ATP, the energy currency of the cell. So what happens? Whatever was happening in part 1, an electrochemical gradient was created, meaning that there is a difference in the electric charge between the 2 sides of the inner mitochondrial membrane, because obviously you have pumped out a lot of protons while the electrons were being relayed along the electron transport chain. So the outside or the exterior of the mitochondrial membrane is positive. Outside means the intermembrane space, the space between the inner membrane and the outer membrane. Now at this point of time, it is positive because of the accumulation of protons, and the inside is negative due to the loss of protons. So there's a electrochemical gradient a chemical concentration gradient that has developed on either side of the membrane. And this electrochemical gradient is how the cell transfers the stored energy from the reduced NADH to FAD and FADH2. So in this step 7, the final step, when there is a high concentration of protons on the outside, the mitochondrial membrane, that is the intermembrane space, the protons are pushed back in through ATP synthase. And this movement generates the PMF, or the proton motive force. This movement of protons causes the ATP synthase to spin and bind ADP and PI, inorganic phosphate, producing ATP, which is the adenosine triphosphate, or the energy currency of the cell. So in summary, one ADP is taken in, one inorganic phosphate is taken in, and ATP is produced. So this is what happens in oxidative phosphorylation. So as we already discussed, oxygen needs to be present. It's a must to receive electrons from the protein complexes, and this allows for more electrons and high energy molecules to be passed along, and this maintains the proton pumping that ultimately leads to ATP formation. So what happens if we run out of oxygen? How do we break down our food to make energy? And we already discussed that question. The body has a plan B, which is called fermentation. It happens all the time in athletes, like runners, when they use all their oxygen and produce lactic acid. And fermentation starts after glycolysis, replacing the citric acid cycle and the ETC, or the oxidative phosphorylation step. And obviously only two molecules are ATP molecules are produced in anaerobic respiration, and either lactic acid is formed or ethanol is formed. There are two fermentation processes: lactic acid fermentation or ethanol fermentation. With respect to humans, the lactic acid fermentation is the one happening. So we'll move on to the next question. 200 finches, 100 pigeons, 50 cockatiels, and 20 sparrows are found in a forest. Assuming there are no other birds in the community, which of the following statement is true? Anybody? It's based on Simpson's index, which was covered. So if you want to calculate, you can. Otherwise, I'll discuss the equation and everything, the meanings and everything. But if you know the formula and if you know the logic, if you want to calculate, you can calculate and let me know the answer. I'll stop for some time. Anybody who's willing to try? If you already know the equation and the, because I think it has already been covered in the lecture. Anybody? Okay, if nobody, I'll take the question. So we'll discuss what Simpson's diversity index is. So Simpson diversity index is basically a measure of diversity. In ecology, it is often used to quantify the biodiversity of a habitat. There is a habitat, there are different species, there are different number of organisms from the different species there. But what's the diversity index? How do we calculate that? And it takes into account the number of species present as well as the abundance, the relative number of each species. So we all know about biological diversity. The name is self-explanatory. It can be quantified in many different ways. The two main factors taken into account when measuring diversity are richness and evenness. Richness means how rich or how diverse the habitat is, and evenness is the diversity even across all the species, or is there like a very skewed schematic? One species particularly much more than the other species? Is there any non-uniform or unevenness, or is the diversity spread across all the species present? So that's what we call evenness. This richness is a measure of the number of different kinds of organisms present in a particular area. Obviously, how many types of species are present in a particular area, that you denote as richness. So it's written, for example, species richness is the number of different species present, obviously. However, diversity depends not only on richness but also on evenness. And evenness compares the similarity of the population size of each of the species present. So evenness is also very relevant because it compares how the population size is like made due to each of the different species present. Like, is one species like contributing much more to the population size, or like all the species are contributing equally? So that's what evenness is all about. Then there's richness. So the number of species per sample is a measure of richness. The more species present in a sample, the richer the sample. Very easy to understand. So species richness as a measure of it on its own takes no account of the number of individuals of each species present. It gives as much weight to those species which have very few individuals as to those which have many individuals. Thus, one daisy has as much influence on the richness of an area as 1,000 buttercups. So very important, very easy to understand. The number is not of importance here. What is of importance is how many species are there, not how many of each species is there. So like if there's species A, species B, and species C. So even if at least one individual of species A is present, one individual of species B is present, and one of C is present, it's very rich in that terms. So it's not calculating that whether there are 10 A individuals present, there are 1,000 B individuals present, there are 1 crore C individual presence. That's what they're not considering in this term, under this umbrella of this term, richness. It's only measuring or only acknowledging the number of different species present. However, evenness is a measure of the relative abundance of the different species making up the richness of an area. So there can be evenness not only depends on the different types of species present, it depends on the percentage, like not percentage, depends on the number of each species that there is in that habitat. So there's an example also here. They have sampled two different fields for wildflowers. The sample from the first field consists of 300 daisies, 335 dandelions, and 365 buttercups. The sample from the second field comprises 20 daisies, 49 dandelions, and 931 buttercups. You can see the table here also. So both the samples have the same richness, exact same richness, because both the samples have three species: daisy, dandelion, buttercup. Okay, and the same total number of individuals. The total number of individuals is also same. In sample 1 there is 1,000 individuals, in sample 2 there is also 1,000 individuals. However, the first sample has more evenness than the second. As you can see the numbers on the table, and you can figure it out on your own why it has more evenness than the second. This is because the total number of individuals in the sample is quite evenly distributed between the three species. In the second sample, most of the individuals are buttercups. There is a huge skewed measurement happening over here because most of the individuals are buttercups with only few daisies and dandelions present. So sample 2 is therefore considered to be much less diverse than sample 1. So a community dominated by one or two species is considered to be less diverse than one in which different species have a similar abundance. So if there's a community that has three species, both the communities have three species, suppose. If one community is dominated by buttercups and the other two types of species are very less, then that makes it less diverse than another community where all the three species are almost present in approximately equal numbers. So as species richness and evenness increase, so diversity also increases. So Simpson's diversity index is basically a measure of this diversity, which takes into account both richness and evenness. I hope the definitions of both these terms are clear because we have discussed now. Now we'll go on to the calculations. So what is Simpson's diversity index and how is it calculated? So it measures the probability that two individuals randomly selected from a sample will belong to the same species or same category other than species. And there are two versions of this formula for calculating Simpson's index. Either is acceptable, but we need to be consistent. Like if we are using only one formula, we need to be consistent with the formula throughout the entire sum. So you can see the formula over here. Small n says the total number of organisms of a particular species, and capital N is the total number of species, all organisms of all species. So this value of D, it ranges between 0 and 1. And 0 represents infinite diversity. Like the closer the value is to 0, that means the diversity is very diverse. It's very rich in diversity. And as the value gets more closer to 1, we can see that it is not that much diverse. The bigger the value of D, the lower the diversity. So next this is not very intuitive and not logical because we have a formula, we put in the numbers and we get. So often there is another term that is used, which is basically 1 minus D. D, whatever the value we get from D, we subtract it from 1, and that is called the Simpson's index of diversity. The Simpson's diversity index and Simpson's index of diversity are different. So what does this 1 minus D talk about? This makes more sense because here in this case, this value represents the probability that two individuals randomly selected from a sample will belong to two different species. So this is the Simpson's index of diversity. The previous Simpson's diversity index is that two randomly selected individuals will have the probability that two randomly selected individuals are from the same species. Simpson's index of diversity talked about different species. There's another way of showing this another term, which is the Simpson's reciprocal index, and which basically means 1 by D. And this index value starts with 1. It's the lowest possible figure, and this figure would represent community containing only one species. The higher the value, the greater the diversity. So Simpson's reciprocal index says that the higher the value, the lesser the diversity. But Simpson's diversity index, which was only D, says that the lower the value, the higher the diversity. So they have given an example here. For example, if there are 5 species in the sample, then the maximum value is 5. Like all the, it will be diverse means all the species will be present in almost equal abundance. So the name Simpson's diversity index is often very loosely applied, and all 3 related indices described above come into the umbrella term. But it is therefore important to know what these terms mean individually. What these terms mean individually. So if you're calculating for this question, in this question there were, the particular question, do you remember, there were 200 where is it, 200 finches, 100 pigeons, 50 cockatiels and 20 sparrows. So for this particular question, if you put the values into the formula, what we see is that there are 4 species, there are 4 types of birds. These are the number of the different types of birds, and we have the formula. So we have done n minus 1. So as you can see from this formula, we get the Simpson's index to be 0.38. That is the probability that 2 randomly selected individuals will belong to the same species. Simpson's index of diversity is the 1 minus 0.38, which is 0.62, and Simpson's index of diversity talks about the probability that 2 individuals randomly selected from a sample will belong to different species. So Simpson's index value is 0.38, Simpson's index of diversity is 0.62. So according to the question, these two answers, 0.6 is the probability that 2 randomly selected birds will belong to different categories. This is correct. This is a Simpson's index of diversity, and 0.38 is the Simpson's index. That's also correct. Moving on to the next question. Assume that the saltwater lake is cut off from the sea. It turns into a freshwater lake over a long time. The jellyfishes lose their sting and microflora changes. This is an example of, anybody?
Succession.
Okay, only succession.
Change in ecosystem. Sorry, it's not succession. It's a
Why not
change in-
Okay, why not succession? Because also it takes over a long period of time. We are not talking about immediate changes.
Yes.
Jellyfishes lose their sting. Jellyfishes are there in the saltwater lake. Now, if the saltwater lake turns to freshwater lake over a long period of time, the jellyfishes are losing their sting. It happens over a long period of time. So why not evolution also? We can call that evolution also. Like jellyfishes are losing their sting.
Evolution should come through only change in the genetic material. That microflora is the different bacteria or algae or fungi. Anything microflora consists of all this.
Yes.
Jellyfish is not that. It is a invertebrate. It is an animal.
Yes,
Almost.
but in a long period of time, it is losing one of its characteristic feature. So
Yes,
why huh?
due to ecological changes, it is not able to survive in the fresh water. That's why it lost its life. It is an entire
Huh.
generation has gone from the freshwater lake.
Yes. So but that, then this is happening over a long period of time. So we, why can't we call that like, if this is happening,
We
why can't we
we
call that
cannot
evolution
we
also?
cannot get microflora from jellyfish. That is
Yes.
not a reverse evolution cannot happen. Evolution can be happen from the primitive to higher species,
Yes,
not from
correct.
higher species to primitive species.
Yes, correct. But like whatever we have learned from evolution, from the Darwin's theory or Lamarck's theory, if there's a particular organism, I am not talking about microflora or bacteria or microorganism in general, any kind of animal organism. With time, if some of their features are lost due to their environmental changes, whatever is happening, and if it's happening over a long period of time, then why can't we call that evolution? They did call that evolution, right? We can, if Lamarck's theory is to be taken into account. Lamarck talked about the long necks of what the animal, giraffe's neck. Why are giraffes neck so tall? So according to Lamarck's theory, apparently at the beginning those were not, the giraffes were not like that, but then they
But
had
in this
to.
case, it is not that because of continuous changes in the genetic material. It is only due to the change in the ecosystem over the long period.
Over a long period of time, huh?
Over a long period of time, it is the genetic material has not got mutated or evolution
Yes,
has
yes.
not happened.
So it's in that case, we like even Lamarck's theory did not specifically say anything about genetic material or mutation. It was also on the outward appearance also. What is theory about the giraffes' necks being tall? Previously, those were not like that. I'm just this is a very discussion, because I am not sure also if we can
That
call this evolution or
if
not.
any part, if it is going to acclimatize the ecology,
Okay.
if any species wants to acclimatize the changes,
Yes.
it is going to change its one or any small parts or extension in the any microbial pathway or biochemical pathway to exist to the ecosystem. If that is going to happen, with jellyfish may come to, if it is a very long period also, it
Huh.
may convert into a fish. It cannot be converted into a giraffe.
Ha
That
ha.
is
That's for
that is
sure.
there.
Huh.
That's why it's not an evolution.
But jellyfishes, they lose their sting. They are not talking about complete different jellyfishes getting like converted to a completely different organism. That's what
Yes.
now they're saying, and they have mentioned over a long period of time. It's not happening overnight. Like what will happen, or like salt water to fresh water. If you're talking about overnight, the first thing that comes to mind, there'll be change in the there will be a hypertonic, basically change in the osmosis happening. The organisms that are very stable in saltwater lake will probably not be able to survive in freshwater lake. That's what comes to my mind if we talk about very sudden changes. But what if they're talking about over a long period of time? The microflora, the microbiota is changing because the environment has changed. Jellyfish is also losing their sting. So this is open to debate. I think this is a very debatable question. So to all their own, I think all of us have very uh, reasons to defend. What I think is that it's definitely a change in ecosystem, but it can also be called evolution, like kind of evolution, but I'm not really sure at this point of time. Maybe I'll go back and read more about it. But to put into perspective, I was thinking in terms of Lamarck's theory of whatever it said about the giraffes, because giraffes did not have those long necks from the beginning, but with time, due to competition or whatever, they needed to, for food purposes, they needed to eat leaves from the tall trees as well. And with time, their necks have become like this, and now they're like this only. It has happened over a lot of, like, it has not happened overnight. It's happened over a long period of time. So with regards to this, I would discuss. So I agree whatever you're saying. I think this is open to debate. So we can all take a little bit time to think about this and maybe discuss this in the next session also. So what does ecological succession mean? Basically, that ecological succession is the process by which the mix of species and habitat in area changes over time, and one community replaces another until a climax community, that's a mature forest, is reached, or until a very huge disturbance like a fire occurs. And it's a fundamental concept in ecology, and this study of succession was pioneered at the University of Chicago by a scientist called Henry Chandler Cowles, who was also one of the founders of ecology as a discipline, as he studied the plants of Indiana dunes. So in terms of ecological succession, we can talk about primary succession, which happens when a new patch of land is created or exposed for the first time. So this can happen when lava cools and create new rocks. Suppose there was a volcano in that place before, there was lava all around, it cools and creates new rocks, or when a glacier retreats and it exposes rocks without any soil. And this is the primary succession. So organisms must start from scratch. So uh, there's nothing there before. So organisms have to start from scratch. So first lichens might attach themselves to the rocks. Then a few small, very small plants might be able to live without without much soil appear. And these are known as the pioneer species, because they are starting off on a barren land, on a new land. They are the first species that are coming up on a new land, and they're starting from scratch. So these are called the pioneer species. Then comes the grasses, shrubs, and shade intolerant trees. These are the intermediate species, and ultimately a mature forest like ecosystem is developed, which is called the climax community. So this happens primary. The word primary is self-explanatory. It happens when a new patch of land is created. Nothing was there before, or exposed for the first time. And there the few small plants or the lichens that come, they are the pioneer species, and gradually the decomposition of these plants, this contributes to soil formation. The soil gets enriched in nutrition, and more larger plants begin to colonize the area. And eventually there's enough soil and enough nutrition in the soil, and to basically enough nutrients become available, and a climax community like the forest is formed. And if this site, climax community is formed, now if this site is disturbed further at some any point of time, that is when secondary succession occurs. So that the name is very self-explanatory. So in secondary succession, what happens? A climax community was already there in that area. It is impacted by any kind of disturbance, maybe a forest fire or anything, any kind of disturbance, any kind of natural calamity or any disaster, and this restarts this cycle of succession, because once there's a natural calamity, everything will cease to exist over there. Any sign of life will cease to exist. There'll be nothing left. So this restarts the cycle of succession, but not back to the beginning, because soil and nutrients are already present. So that's why this is very different from primary succession, because nothing is starting from scratch. There was something already there, it got destroyed, and now it's going back to the beginning, but with soil and nutrients already available and present. So, for example, they have given the forest fire example only, that kills all the mature trees on a particular landscape. Then some grasses might start growing, followed by shrubs and a variety of tree species, until eventually the community that existed before the fire is present again. So this is called secondary succession. So this is how secondary succession happens. There was already some kind of community, some kind of ecosystem present there, but due to a forest fire, natural calamity is what generally hinders this kind of community, and then this entire thing is blown away, but there's soil and nutrients already present, so nothing starts from scratch, but everything falls, everything, there is a cycle of succession, everything restarts again, and we reach another climax community that was very similar to what was already present with time. So this is called secondary succession. So an example of ecological succession, so there's many in many different contexts, I think, and in many different time spans, many different people have come up with different examples. So in Hawaii and Iceland, primary succession occurs on lava flows when new land has formed. So there was lava flow, new land has been exposed, and that's where primary succession happens. But in Canada's Athabasca dunes, it happens when new sand is deposited along a lake shore. In the Andes, it occurs when glaciers retreat. So these are all examples of primary succession. But in many regions, secondary succession also occurs when wildfires have destroyed conifer forests, or where former agricultural land is reverting to meadow or scrubland. So what these examples have in common is that the climax community is not the first present on the landscape after succession begins. First, intermediate communities occupy the space, sometimes for many years, creating ideal conditions for the communities that follow. The mature forest, the mature climax community doesn't happen overnight. It takes time for that environment, for that ecosystem to be alive again in case of secondary succession. And we are talking about climax community a lot. So what is a climax community? Climax community is basically the end point of succession within the context of the particular climate, context, the particular environment, with whatever is available, geography and everything. And in the for example, they've given in the Midwestern US, for example, such a community might be a hardwood forest with oaks and hickories as the dominant tree species. So this climax community will persist in a given location for a very, very long time. It doesn't happen overnight, and it doesn't get destroyed overnight. It can get destroyed overnight in case of any natural calamity or disturbance, but it takes time for this thing, climax community, to come, years and years, and this will stay in that location. There will be not further progression from that until a disturbance occurs, like forest fire or anything. However, in many ecosystems, disturbance occurs quite frequently enough that a matrix of community types may be consistently present in the landscape. So, if in any kind of environment, disturbances are actually very likely to occur very often, then a matrix of community types forms, where all the time there may be some kind of community, climax community already present on the landscape. So, for example, they have given in an area prone to wildfires like the western US, mature forests may exist near grassy meadows with fewer scattered trees, and consistent disturbance and variation in factors like water and nutrient availability over the course of decades. This allows many plant and animal communities to thrive within a particular climate and geographic niche, not just those adapted to the absence of disturbance seen in the climax communities. So I think what climax community is very clear. You can see from the images here also. Climax community is basically a mature forest, very relevant and very conducive. That's very conducive for growth in that particular environment. So we know what an ecosystem is. A geographical area where all biotic and abiotic components interact with each other is called an ecosystem. And these ecosystems are dependent on a lot of factors, environmental factors. Also, any factor, if going through a massive change, can cause a change in the ecosystem. These changes might be climate, habitat, invasion, pollution, invasive species, overexploitation, etc. And when most of these environmental conditions change, the kind of animals found here will also start changing. So newer species will substitute the former ones. The former ones were adapted to a particular type of environment, but with environmental changes, if the that kind of species is not anymore able to survive over there, the newer species will substitute the former ones. Former ones will not survive anymore. There will be newer species which will be whose growth is more conducive for the environment. Survival of the fittest, as we know. So in ecological succession, some in a barren area, some species that are observed first will be the pioneer species, and this kind of species will make the soil softer and more adaptive for other species. There'll be lichens or everything that will make the soil softer and further they'll make the soil very conducive for the growth of the mature forest. And after pioneer species, there'll be intermediate species like shrubs and grasses, and then after intermediate species, large species will start dominating the area called the climax community. And after the starting the climax community, it can't be substituted easily. So the climax community is kind of like the end point till a disaster strikes. So after the establishment, there cannot be further progression from that. And food chains, food waves, and energy flow, everything will be observed in that particular ecosystem. And these changes will occur gradually, and new species will be introduced. So this is called ecological succession. Why I thought that the example that we had in the question might also be ecological succession, because at some point of time the salt water got changed into fresh water, and then with time new species are introduced. The previous microflora is not there. New species, new organisms are now introduced. So this can also be an example of ecological succession according to my point of view. Uh, this can also
Yes.
be termed as an
Yes,
ecological.
that's right. As you rightly said, that's right. But as per regarding to the jellyfish, only string, losing the string is maybe due to this succession or evolution.
Okay.
But whereas in the ecological conditions, that exosmosis will come and the entire the jellyfish will die
Ha
in
ha.
that ecological
True.
condition.
True. Yes.
That is the
That
reason. That's why
yeah. That's what will happen in the first case. Like you're shifting a organ. That's what I said. When if sudden thing is happening, the first thing will happen because there's a salt water and there's fresh water. So there's solute concentration will matter, hypotonic osmosis, everything will matter, and jellyfish will not survive at all at the first chance.
yes, that's right.
It'll not survive because it survives in salt water, and you're putting it in fresh water where there's no salt or salt concentration is not there. Then obviously there'll be very it will not survive basically. So that's a sudden change, definitely.
Yes.
Agree. So ecological succession, it occurs in 5 stages: nudation, invasion, competition, coaction, reaction, stabilization. So a forest is destroyed due to fire. Now on the dead organic matter, microbes will grow and decompose it. These will be called the cellule. And now the soil will become more fertile and suitable for plant growth. The small shrubs and everything that will follow, and then the terrestrial plants will grow in the majority of shrubs, and then the woody plants that will give rise to the climax community. Next, we'll move on to the next question. Which of these is not true with respect to extracellular polymeric substances? It creates a mesh and traps cells of the biofilm. It entraps nutrients for the cells, gives protection to the cells in the biofilms, and toxin production to protect biofilm from invasion. Anybody would like to try? Okay, so I'll talk a little bit about extracellular polymeric
Option
substances.
fourth.
Option 4, you think it's not true? Okay, yes, you're correct. But we'll talk a little bit about extracellular polymeric substances first, and then we'll go through the options and see why the first 3 options are true, mostly true. So one of the key elements in the establishment and maintenance of biofilm structure and properties is the extracellular matrix. So in nature, basically bacteria alternate between 2 modes of growth: a unicellular bacteria, a unicellular life phase in which the cells are free swimming planktonic, and a multicellular life phase in which the cells are sessile and they live basically in a biofilm that can be defined as surface associated microbial heterogeneous structures comprising different populations of microorganisms surrounded by a basically self-produced matrix that allows their attachment to inert or organic surfaces. So while a unicellular life phase allows for bacterial dispersion and colonization of newer environments, biofilms allow sessile cells to live in a coordinated, more permanent manner that favors their proliferation. And in this alternating cycle, bacteria accomplish two physiological transitions via differential gene expression, from planktonic cells to sessile cells within a biofilm, and from sessile to detached newly planktonic cells. And many of the innate characteristics of biofilm bacteria are of biotechnological interest, such as the synthesis of valuable compounds such as surfactants, ethanol, and the enhancement processing of certain foods such as table olives. So, understanding the ecology of biofilm formation will basically allow the design of systems that will facilitate making products of interest and improving their yields. So this is basically the all the features of a biofilm. So what happens, the extracellular matrix of the biofilm structure is comprised of water and extracellular polymeric substances. This is what is EPS, which are basically polysaccharides, proteins, and even DNA. And characterization of the matrix requires component identification as well as determination of the relative concentration of these EPS constituents, including their physicochemical properties and descriptions of their interactions. And biofilm is basically a community of aggregated cells organized as microcolonies living at an interface between a surface and a liquid phase and embedded in an organic polymer matrix of microbial origin. Bacteria, extracellular polymer, and bacteria surface interactions, all these drive the formation and behavior of biofilm. And these EPS or extracellular polymeric substances are basically organic polymers of microbial origin and involved in the bacterial cells' interactions with their environment. These are formed of polysaccharides, proteins, extracellular DNA, and lipids. And within biofilms, EPS are distributed between the cells in a very non-homogeneous pattern. And these helps the cells to interact with each other, and the matrix that is formed, it encompasses all the microbial cells. And the stability of this matrix is ensured by non-covalent bonding between these extracellular polymeric substances that involves weak physicochemical forces. And this network that interacts with the bacterial aggregates, it confers basically cohesion and viscoelasticity to the structure. So what is the role of the different classes of EPS in biofilm behavior? It can be studied through various experimental approaches. So these some researchers, some studies are basically very actively working on the role of this extracellular polymeric substances in various in the biofilm behavior. How does the biofilm behave? So this is basically all the features of the biofilm. So there's genetic exchange that have this drought protection. They can aggregate this adhesion cohesion. It helps in adhesion cohesion. It helps in gaining symbiotic relationship with plants. This biofilm structure, it can trap nutrients in a mesh-like environment, basically for the goodness of the bacterial cells. There's a pathogenicity or virulence factor as well. It engages in signaling. We have all heard of quorum sensing part, so that's kind of signaling only. Quorum sensing is a very popular term that's used. It helps in that. There's a genetic exchange. It uses carbon source. So all of these is what makes biofilm very fascinating, and many researchers are actively studying it. So yes, it creates a mesh and trap cells on the biofilm, definitely true. It entraps nutrients for the cells, also true. And it gives protection to the cells in the biofilm. Obviously they are staying in aggregate form, they're staying in a very aggregated manner, and that is what confers them a different kind of protection than when they're in the unicellular planktonic phase. Moving on to the next question, which of these is true? Microbial community gradient is inverse to the electron acceptor gradient. Energy yield decreases as the redox potential decreases. Sessile bacteria attach to a substrate and produces the cellular polymeric substance for further attachment. Microbial mats are layers of microbial guild. What do you think? Which ones are true? Anybody would like to try? Take your time.
Option C.
Option C is true. Any other? Correct. Any other thing you think are true? Option C is not mentioned. The option is also true. Sessile bacteria, it basically not attached to a substrate. Basically, sessile bacteria is it's basically like as I said before also, that bacteria basically alternate between two modes of growth. So the multicellular phase will be the sessile bacteria. They basically live because they are in aggregation. They live, they're sessile because they live in a biofilm, and that can be defined as surface associated microbial heterogeneous structures. So attached to substrate, I think they produce all the bacteria, they ultimately lead to the production of EPS. So yes, the cell bacteria attached to substrate that can ideally that should also be true. Microbial mat, I'll talk a little bit about what are microbial mats. And then energy yield decreases as the redox potential decreases. That is also true. I'll explain why one by one. So anaerobic decomposition of biomass to carbon dioxide and methane only occurs in anoxic environments, and it happens with a very low redox potential where concentrations of other electron acceptors including nitrate or oxidized forms of metals such as manganese or ferrous or sulfate are low. So the inhibition of this anaerobic digestion by nitrate, oxidized metal ions and sulfate is determined by this redox potential bar. And a decrease in the redox potential leads to changes in the dominant type of anaerobic respiration towards low energy yielding processes. So the nature of the final electron acceptor present in the environment is a key factor in determining the ecological niches for particular microorganisms. So we all talked about this, about the key, about the electron acceptor, how in oxidative phosphorylation it's oxygen, how in other cases it's not. So based on all of this, we can say that the nature of the the terminal electron acceptor that there is, will play a very determining role in developing the ecological niche for the organism. So that option is true. Energy yield increases, the redox potential decreases, as you can see from the energy yield here also, because oxygen will give rise to the highest yield, highest energy, as you can see from here. Next, we'll talk about sessile cells, the second option we were talking about. So as I was talking about, the bacteria alternates between two living life forms, the planktonic freely swimming cells and the sessile cells. So the sessile cells, how does this transition happen? So the transition from sessile to dispersed cells and returning to the planktonic cells, because the biofilm dispersed cells were transcriptionally closer to biofilm cells than to the planktonic cells, but with several differences. And this extracellular matrix that the sessile cells finally form, this is what helps in their technique of quorum sensing, which is nothing but signaling. They are actually moving in an aggregate structure, and the signaling process, the way they're interacting with each other, is called quorum sensing. So as you can see from the sketch here, this is the unicellular life phase of the planktonic form. All the bacteria are separate, then there's attachment starting, there's resorption, then there's a maturation phase where the multicellular life phase or biofilm is formed, and then at some point of time there is detachment happening where the sessile bacteria gets converted back to planktonic bacteria. So sessile bacteria option is also true. Then next we'll come to the third option, which was microbial mats. So basically, what are microbial mats? Microbial mats are basically considered to be extremely thick biofilms, and for example, in case of chemolithotropic and phototropic bacteria, these also build these kind of microbial mats. And microbial mats form layers that are composed of different microbial guilds whose activity is dependent upon the amount of light available and other resources like nutrients. And different organisms are living in the mat, and the order in which they are layered, it's a very thick layer of biofilm, right? The different organisms can live in this mat, and the order in which these are layered depends on the nutrient transport controlled by diffusion. And these various microorganisms form the layers that make up the microbial mat. And in today's day, microbial mats are quite declining in abundance due to the competition of aquatic plants and metazoan grazes. Today, I think microbial mats develop only in the marine environments where competition is still restricted, and these environments are very hypersaline or geothermal habitats, because microbial mats can grow only in extreme conditions and remote locations. It's difficult to study them today, and that's the reason it's very difficult for researchers to study them today. And these are very sensitive to changing light intensity. Light intensity significantly influences the chemical and biological structure of a microbial mat. So light intensity actually plays a very huge factor in influencing the chemical and biological structure. That might also change the kind of organisms that live in the different layers. And I think cyanobacteria is the most abundant and versatile microbial mat builder, because they can grow in hot temperatures or cold temperatures. They're versatile. These cyanobacterial mats contain large numbers of primary producers. So these primary producers, what do they do? Primary producers use light to synthesize new organic material from CO2. And cyanobacteria, unlike microbial mats, grow in places that are easily accessible to scientists, like the Yellowstone Spring or other thermal regions of the world. So it's easiest to study in that way. Then there are chemolithotropic bacteria also, microorganisms which also build mats, and these mats are comprised of filamentous sulfur oxidizing bacteria which grow on the marine sediment. And the chemolithotropic mats oxidize hydrogen sulfide or H2S to support energy conservation and autotrophic reactions. And the microbial mats on Earth composed of the Theoploca species. We talked about the Theoploca species in one of the previous sessions. There are microbial mats composed of this Theoploca species on sediments of Chilean and Peruvian continental shelf, and these are thought to be the most extensive microbial mats on Earth. So these are some of the information that I gathered from research papers. If you are interested, you can also go and search. And these microbial mats basically start as biofilms, but as biofilms form on a surface, they become more and more complex. They develop layers and layers. They become very thick and diverse, and the diversity reaches its maximum, and that's when we call it a microbial mat. And but microorganisms do not build microbial mats just like that. They need energy and water to build it, and water can be provided by hot springs, lagoons, or even a coastal shoreline. And many microorganisms use solar light also as their main energy source. So with enough energy and water, microorganisms can easily flourish on a surface. They stick together with food and grains of sand or soil, and building mats that can be up to few centimeters thick, if you can see the sketch over here. And in some cases, a new living mat grows on top of an older dead mat, creating thick layers. And as the name suggests, mats resemble carpets or rugs in a way that they cover surfaces of various sizes. And when seen up close, mats also have interesting vertical layering, as you can see from the sketches over here. And they can be distributed in green, orange, red, purple layers, with each layer representing a different community of microorganisms that need different amounts of sunlight and oxygen. Obviously, the microorganisms in the deepest layers will not require that amount of sunlight to survive. Otherwise they would not be able to survive in the deepest layers at all. So they have chosen their habitat. So they obviously this is they do not require that kind of sunlight to survive. And all the microorganisms in the mat work together in a synergistic manner to sustain themselves and to interact basically with the surrounding environment. These are what microbial mats are. So studies have shown that microbial mats are important for the functioning of ecosystems, and a community of microorganisms living in a certain area and the non-living components of the environment is what is called microbial mat. So for example, they have said that when mats colonize the ground, they contribute to the health of soils and sediments, producing nutrients that enrich them. And mats also participate in the recycling of some chemical elements, including nitrogen, carbon, and sulfur. They can fix some of the chemical elements. They can also clean the water. They can both take up and release gases from the atmosphere, such as oxygen, hydrogen, carbon dioxide, and methane. And mats are also good food source for animals. Some flies, snails, worms, crabs, and birds feed on small pieces of microbial mats, and larger organisms can then feed on those smaller ones. So it's basically a food chain that is happening because they are continuously eaten. Mats often do not grow much except in extreme environments. In extreme environments, when their higher level organisms are not present, maybe because they cannot survive over there, and in those regions, mats might show enormous, tremendous growth. Otherwise, they are constantly eaten, so they do not show that kind of growth also. Next question is, the answer is already seen over. Okay, anyway, weather conditions suitable for persistence of flu virus in aerosols. So you can already see the answer, but did anybody have any different opinion or so? Do you want to still take a guess, and do you have a conflicting opinion? Anybody? So I'll explain basically why the temperature for flu virus is quite fascinating, because there are also papers published because people have actually worked on it and they have published their data. So if we're talking about flu, let's talk about the influenza virus, which is a virus transmission that is tremendously dependent on the transmission is tremendously dependent on humidity and temperature. So in one such paper, scientists have actually used a guinea pig model to check the influenza virus transmission. So they tested directly the impact of ambient temperature and relative humidity on the efficiency of viral spread between the hosts. So they inoculated and exposed the guinea pigs, and when these inoculated exposed guinea pigs were housed in separate cages, like there is no physical connection, there is no physical way where they can touch each other, transmission was then found to be dependent on both temperature and relative humidity. And transmission was highly efficient at 5 degrees Celsius, but was blocked or inefficient at 30 degrees Celsius. If you can see the graphs over here, the blue ones show this is from the paper, the blue ones show the 5 degrees Celsius. So transmission was highly efficient, as you can see from here. But at 30 degrees Celsius, transmission was not that good or inefficient. And this is in case of relative humidity, and this shows the absolute humidity. Dry conditions like 20% and 35% relative humidity were also found to be more favorable as you can see, for growth, for spread, then either intermediate like 50% relative humidity or 80% relative humidity. So all these results were obtained initially using a particular strain of influenza virus and then was subsequently confirmed with another different pandemic isolate strain from Netherlands. And transmission at low versus intermediate temperatures was also tested with two influenza B viruses, and they were found to be more efficient under colder conditions. This proper research work has been conducted. The transmission of human influenza viruses by a respiratory droplet or aerosol route in the guinea pig model proceeds most readily under cold, dry conditions. And these findings suggested two means by which environmental factors may drive the wintertime seasonality of influenza in the northern hemisphere. So they've seen average daily temperatures outdoors are lower in the winter, and conversely, average outdoor relative humidity is higher in winter than in summer, essentially excluding outdoor relative humidity as a possible driver of influenza outbreak. However, because indoor air is commonly heated, indoor relative humidity is lowest during the winter. Thus, exposure to cold air outside or dry air inside during the winter time may increase influenza virus transmission and potentially also trigger a flu season. And they have tried to actually match various mechanisms to the data that they have found. That is what they have done. They have conducted experiments, they have generated data, then they have tried to put in the context of previous research, and they have tried to hypothesize some of the possible mechanisms that might be linking temperature and humidity transmission. So it remains unclear precisely why influenza virus transmission is most efficient under cold and dry conditions, but a number of possible mechanisms have some experimental support. So through they have they have basically through nebulization of virus into a chamber, followed by serial sampling and then determination of infectious titers, influenza virus stability in aerosol was shown to vary with relative humidity or absolute humidity, given that at that point temperature was held constant. So it was seen that influenza virus was consistently found to be stable at low relative humidity and relatively unstable at intermediate relative humidity. And studies differ in the results seen at higher humidities. The stability was low in some reports and high in some other. And they conducted a more recent examination of the problem that reconciles the findings at high humidity by demonstrating a dependence on salt and protein concentrations within the droplet media as well. So the authors of that report furthermore hypothesized that the effect of relative humidity on virus viability is mediated by salt concentration within the droplets. Like at higher relative humidity, physiological concentrations are maintained and viruses are relatively stable. At intermediate relative humidity, evaporation leads to increased salt concentrations, resulting in virus inactivation. And at low relative humidity, salts crystallize out of solution, yielding low salt concentrations and high virion stability. So taken together, all these reports, they have suggested that improved stability of influenza virus within aerosols may account for the enhanced transmission seen at low ambient humidity. So they have shown that at low ambient humidity, there is the viruses stay more stable in the aerosol droplets, and that's why the transmission efficiency is also more. And these viruses are also known to be more stable in the cold. So as you can see, robust transmission is happening at 5 degrees Celsius and highly inefficient transmission is happening at 30 degrees Celsius. It may be due to an increased virus half-life at lower temperatures. Virus half-life, they have hypothesized that it increases at lower temperatures. And in considering the effects of temperature, it is important to note that guinea pigs shed higher titers of influenza A and B viruses when housed in the cold. So when they are kept in the cold, they actually shed more titers of influenza virus. And this increased shedding may be due to the effect of cold conditions on the host. Alternatively, virus may be more stable within the nasal passages when the epithelial surface is cooled by colder ambient air. And there is an increased level of virion stability at low temperatures, likely due to the part of decreased activities of proteases. So there's decreased activity of proteases all around. That's why virion stability is really much more in cold regions. In addition, changes in the physical properties of the virion and envelope may contribute. At temperatures less than 40 degrees Celsius, some domains of ordered and disordered lipids are found to coexist within the virion membranes. And this fraction of lipids within ordered domains or a gel phase increases with decreasing temperature. So lipid ordering can also be linked to the infectivity. So in addition to having an effect on the virus, this relative humidity and temperature may also be having an effect on the host. Some kind of some range of temperature, some range of humidity might be making the host also more susceptible, host also more vulnerable to the virus infection or the course of disease following the infection. So cooling of the nasal epithelium through inhalation of cold air. So during the cold, we are inhaling the cold air, and the nasal epithelium gets cooled. So this has been shown to inhibit various mucociliary clearance and may limit the phagocytosis by innate immune cells resident in the upper airways. So there are innate immune cells that are present in the in the upper airways. So this inhaling of cold air might inhibit this mucociliary clearance, and the phagocytosis that happens when their innate immune cells basically engulf foreign pathogens. This process can also be restricted by inhaling of cold air. So similarly, inhalation of dry air for a 30-minute period was found to show mucociliary clearance significantly. So both cold air and dry air are thought to be like alter the rheological properties of the mucus, and at lower temperatures cellular metabolic functions are slowed, which in turn may decrease the frequency of ciliary beads, reduce mucus secretion, and restrict phagocytosis. So finally, all these environmental conditions, all this can impact transmission through effects not only on the vehicle itself but also on the respiratory droplet, also on the host which is taking in the virus, and the length of time that the virus droplet stays airborne and therefore available for inhalation is also dependent on the size. So how the evaporation of water happens to respiratory droplets, which occurs more rapidly with declining relative humidity, because it decreases the droplet size and therefore increases the distance and time over which transmission can occur. So that is how they have tried to hypothesize that cold and dry conditions will impact the stability of influenza virus particles and the innate defense of host nasal epithelia and the production of infectious bioaerosols. So each of this effect contributes a very plausible and believable explanation for the impact of relative humidity and temperature on this respiratory droplet transmission, and more than one mechanism most likely contributes to the observed transmission outcomes. So one is that host becomes more susceptible because the phagocytosis here in the respiratory airway might not be working properly. The virus is more stable because of lesser function of the proteases all around. So all these, all these might be responsible for more transmission during cold and dry weathers. So as I already discussed, the two physical properties are relative humidity and absolute humidity, and this is a very relevant parameter for considering the rate with which water will evaporate, how the respiratory droplets will form. So this is a very complex means of describing the water content of air. So I've already described the role of this thing. So this that's why weather conditions that are suitable for persistence of flu viruses and aerosols will be either hot and humid or cold and dry. So next question is choose the options which are true. This also is it's easily seen. You can see the options, but does anybody have any conflicting opinion? The answer is already visible. Does anybody think something else, or do you have any kind of different opinion? According to me, all of these options are true. I'll explain one by one, but anybody else? I'm still in the meeting, right? So I'll move on to the answers. I'll describe why I think all these options are true. So we'll first talk about biological oxygen demand. So I think we have already discussed this um, the meaning of biological oxygen demand in the first class. So it's nothing but it represents the amount of oxygen consumed by the bacteria and other microorganisms while they decompose organic matter under aerobic conditions, where basically oxygen is present. That is what is called the biological oxygen demand, and at a specific temperature. So drinking water has a BOD, or the biological oxygen demand level, of 1 to 2 ppm. When BOD value of water is in the range of 3 to 5 ppm, the water is moderately clean. Polluted water has a BOD value in the range of 6 to 9 ppm. In polluted water, some organic waste is present. So you can see, higher the BOD value, more chances of organic waste being present in that particular water. So you can see on the sketch over here is that there's white is oxygen. More and more is oxygen is being consumed by the microorganism present in the water. That leads to the demand for oxygen, which is here schematically shown as white circles inside the big blue circle. And more and more organisms if present in a particular water sample, there will be more consumption of oxygen, and then the demand for biological oxygen will be more, and bacteria will utilize more and more oxygen, and they consume, and this oxygen will get depleted in water. So that is what has been shown here. That is what biological oxygen demand measurement happens. That is how it happens. One second. This entire thing happens. Sorry, there's some internet issue. Okay. So the significance of biological oxygen demand is that it is a measure of the amount of dissolved oxygen that is being used up by microorganisms as they break down organic matter in water. And the higher the biological oxygen demand, the more oxygen that is being used up and the more polluted the water is. So this makes biological oxygen demand a very important indicator of water quality. So you can see from here, the increase in BOD can be caused by higher microbial load. Very true. Higher the microbial load, more the number of organisms present in that particular water sample, more oxygen is being consumed by the organisms, and hence the biological oxygen demand is increasing. So increase in the BOD is basically can be extrapolated to be caused by higher microbial load. So the BOD value that is determined using microbial respiration, it can be affected by amounts of dissolved oxygen in the sample. It is known that some synthetic compounds, such as artificial electron acceptors, can be reduced by certain microorganisms, like artificial electron acceptors. And if these compounds possess reversible redox properties, they can serve as carriers of electrons from the biocatalytic systems of microorganisms to the electrode. And when using these mediators, the results of measurements become in practice independent of the partial pressure of oxygen in the medium. And if the oxidation of the reduced mediator does not involve protons, the mediator electrode can relatively insensitivize to pH changes. Thus, one of the most promising trends is the development of BOD sensors using electron transport mediators. So BOD measures the concentration of electron acceptor, kind of also works out for that. So next, the other question was mobility of microorganisms increases in nutrient-rich water. So as we know, uh, motile bacteria can use the process known as chemotaxis, where they basically understand chemical signals to respond to the nutrient gradients associated with the enhanced dissolved organic matter concentrations within the phycosphere. So in that case, motile bacteria are using chemical signals to respond to nutrient gradients. So yes, that in that sense, if we think with that perspective, the first option is also true. Mobility of microorganisms increasing nutrient rich water. In nutrient rich water, they'll respond to the nutrient gradients, and they'll henceforth be more motile. Hello, really sorry guys, there was some network issue due to which I was thrown out of the meeting. I'll share my screen once. One second. So you don't have to worry, I'll upload this video on YouTube. So even if you have missed a part, I actually kept talking and then I realized that I was not in the meeting. So but that part is recorded, so I'll upload that part to YouTube, so you can check that out and maybe we can discuss in the next session if you have any questions after watching the video. So I'll share my screen. I think it's visible. Yes, so we'll move on. This question was already discussed, how temperature, the flu virus, the kind of temperature that is conducive for flu virus growth. I've actually explained in very detail. So I think you have heard, I think till the middle of it. The rest of the portion I have already recorded. It will be uploaded on YouTube. So this is the next question. Which of the following options are true? Mobility of microorganisms increase in nutrient-rich water. Increase in BOD can be caused by higher microbial load. BOD measures the concentration of electron acceptor. Biofilms are likely to develop in lakes with algal bloom. Anybody? Anybody would like to try? Okay, I'll discuss
Option B.
Option B is true. The other options are not true. Okay, so
Yes, yes.
we'll talk a little bit about the options first before going to the answer. Wait one second, my laptop is. Wait one second. Yeah. So what is biological oxygen demand? I think we discussed biological oxygen demand in the first class only. So it's basically the demand of oxygen that is created in any kind of sample, as and when more and more oxygen already present in the sample are getting consumed. So if there's a microbial load is more in the sample, like if there are a lot of microorganisms in the sample, they'll be taking in more and they'll be consume more and more oxygen, and that will show a readout as oxygen demand. So if you can see in the sketch over here, this is a very rough sketch. It is shown the oxygen is shown in white circles, and this bacteria, if there's more and more bacteria, the microbial load is more and more oxygen will be consumed, and there will be an increasing demand for the oxygen in the sample then from that point onwards. So that has been shown like in gray color. The gray circles mean BOD. So if obviously if there's more BOD demand, biological oxygen demand in a particular water sample, that means most of the oxygen has already been consumed. That means the microbial load is very high. So that is why drinking water, which we basically drink and we get inside our system, that has a BOD level of 1 to 2 ppm, which is very low, and that is why drinking water is considerably comparatively pure. When the BOD value of water is in the range of 3 to 5 ppm parts per million, the water is moderately clean. But polluted water will have BOD value in the range of 6 to 9 ppm. 6 to 9 ppm BOD value means there's a huge oxygen demand. That's because the oxygen already present has already been taken in by the consumed by the microorganisms present in the sample. So it shows that the microbial load is very high in polluted water. So that's why biological oxygen demand talks a little bit about the higher microbial load. That is true. Increase in BOD can be caused by higher microbial load. So significance of BOD, as we discussed, is that it's a measure of the amount of dissolved oxygen that is used by microorganisms as they break down organic matter in water. So the higher the biological oxygen demand, the more oxygen that is being used up and more polluted the water is. This makes BOD a very important indicator of water quality. Also, the BOD value can be determined using microbial respiration. That can be affected by the amount of dissolved oxygen in the sample. So it's known that some synthetic compounds such as artificial electron acceptors can be reduced by certain microorganisms, that is are artificial acceptors of electrons. Now if these compounds possess reversible redox properties, they can serve as carriers of electrons from the biocatalytic systems of microorganisms to the electrode as well. So when using mediators, the results of the measurements become in practice independent of the partial pressure of oxygen in the medium. And if the oxidation of the reduced mediator does not involve protons, the mediator electrode can be relatively insensitive to the pH changes. So one of the most promising trends in development is of BOD sensors using electron transport mediators. So another option also gets checked out. So this is from a paper. They are working on BOD sensors that are relevant to electron transport mediators. Then the first option was mobile bacteria increases motility in nutrient-rich medium. I would say increase, but there is a change. There might be change in bacterial motility when there is in the nutrient-rich medium. So as we know that motile bacteria use the process called chemotaxis to basically respond to nutrient signals, nutrient gradients. So it's basically a type of kind of signaling. They're reacting to chemical signals that are in the their immediate surroundings. It's called chemotaxis. They respond to the nutrient gradients associated with the enhanced dissolved organic matter concentrations within the phycosphere. So motility of bacteria might change in response to the nutrient gradients. It's basically they change the direction in response to the nutrient gradients. So in that perspective, that option also checks out. Another question was that can biofilms be seen in the presence of algal blooms? So what are algal blooms basically? We need to know that. And what are biofilm growth? We have already discussed biofilm, sessile bacteria, and all that. We already discussed in one of the questions, but we'll have to discuss what algal blooms are. So anybody would like to take a guess what algal blooms are? You might have seen them everywhere. They are very they can be very easily seen in a humid atmosphere like in our country. But algal bloom is not just a very smelly scourge. It is a presence of algae, and it leads to much toxic concerns in the future. That's what happens in the presence of algal blooms. There's increased risk of toxicity of recreational water to people. The farm pond water is very toxic to livestock because livestock generally get their quench their thirst from farm pond water. There can be pH imbalance due to the rigorous treatment methods needed to regulate algae growth. So there's a lot of algae taking up the entire fish pond. So there'll be a lot of treatment also that the farmers will do to regulate the algae growth, and those kind of treatment may also cause a very disastrous pH imbalance. Then there will be fish kills due to the algae die of hatcheries, lakes, and private ponds. The fish in the pond will get killed due to the algae. Then the algae on their own will be a rather difficult thing to remove. Because it is often accompanied with the growth of biofilm, that is bacterial aggregation, that slimy growth of bacteria that tends to cling to solid surfaces present in water. Then there'll be swimming safety concerns. People will not be able to swim properly. There'll be boat propeller entanglement. There'll be increased water treatment, operating costs for clarification, filtration, taste, etc. Reduced property values due to the sight and smell of algae. Financial loss. There will be increased risk of red tides, where polluted rivers and streams will meet tidal and marine waters. So algae on its own can be a rather vexing issue, but often with biofilm it becomes really, really toxic. And added concerns from biofilm growth in water also shows increased maintenance and cleaning costs. There will be a potential generation of carcinogenic chlorinated byproducts such as trihalomethanes and haloacetic acids, which, if not controlled, can lead to heavy fines and penalties. There'll be heat exchanger failure and loss of cooling efficiency, both of which will drive up operating costs. There'll be under deposit corrosion in piping and both surfaces. There'll be manganese precipitation, and it will be notable for a change of color on the surface of heated water. So all this often dismissed as ponds come, this algae and biofilm, these are real villains of the story, and they can actually pretty much coexist together. They are very complex organisms that can cause havoc for water ecosystems and the humans that rely upon. So this green enemy that would otherwise be distorting the landscape and negatively impact our senses, including sight, smell, and they are defined as a group of basically predominantly aquatic, photosynthetic, and nucleus bearing organisms that lack true roots, stems, leaves, and specialized multicellular reproductive structures of plants. So basically, they are not just one insidious creature, they are a host of organisms that may vary based upon location, but share the same basics in common. And with that, with biofilm also, these dense colonies of bacteria, they love to stick together through thick and thin, and they'll also give rise to this biofilm. And like biofilm removal cost, it's very high maintenance, keeping these ponds clean, the cleaning cost, the everything. Then the creatures and the carcinogens. Biofilm in water can promote other space invaders such as barnacles, then mussels, oysters, and some of this, the essential byproducts can also include manganese precipitation, as I already said before, and various harmful carcinogens. So bacteria will very easily exist in biofilms which are surface adhering, and then free floating groups of cells with the extracellular polymer matrix. So there's a easy cooperation between algal bloom and bacterial biofilm. So even that option is correct, that algal bloom, biofilms are likely developed in lakes with algal blooms. Yes, it can happen very easily, and that will lead to a lot of risk. So these I've already discussed. This is the next question. We can discuss this question and then maybe move on, or we can discuss the next question. Wait, this question we can discuss. Choose the relevant option. Piezo-tolerant microbes can be found in deeper oceans when compared to piezophilic microbes, and some bacteria can oxidize and reduce sulfur. Which one do you think is true? Or are both true?
Only two is true.
Okay.
Only second one. It's true.
Yeah, correct. Because piezo tolerant is very self-explanatory. Piezo tolerant, piezophilic is to fill is to love. So and piezo is basically related to pressure. So piezo tolerant is like they are tolerating, the microbes are tolerating the pressure, but those microbes cannot be found in much more deeper oceans than piezophilic microbes. So I found this graph from a paper that show that a piezophilic is an organism that lives under elevated hydrostatic pressure, while piezo-tolerant organisms only tolerate high pressure. They can tolerate and live under high pressures, but piezophiles are organisms that grow better in high pressure. And strict and obligate piezophiles require pressures above atmospheric pressure for growth. So you can see the hyperpiezophiles will grow really well with increasing pressure. The piezotolerant valves, they can also grow well in greater pressure. Piezophiles will be very happy to grow under pressure, but the piezosensitive bacteria and archaea, their rate will start decreasing after a certain point when much more pressure is applied. So that is how this trend works on. So this is the last question for today. I know in the middle I got actually thrown out of the meeting, but I'll upload the entire thing on YouTube so you can check that out. And if you have further questions, maybe we can discuss. We can discuss the evolution and succession question in the next session. Also, one of the green sulfur bacteria question that we had a doubt on, which is most likely to be found in hot spring, green sulfur, purple sulfur, that question. We can also read more about it and maybe discuss why these which kind of bacteria are more likely to be found in hot springs. So if anybody has any question, we can take those, or else I'll end the meeting. I'll upload the recording on YouTube and maybe we can conclude the session for now. Anybody? Okay. So if you don't have any questions, we'll conclude the session today and we can maybe discuss some of the remaining questions in the next session, and we'll also discuss the assignment questions after the due date is over. So everyone, you can leave. I'll stop the meeting. Thank you so much, everyone, and sorry for the technical issues. I'll close the meeting.